let's get started. So last time we have finished with this nice looking guy, so with the bear, and today we are moving further. So we have a little bit what to say regarding the usage of fats for getting energy, and then we will get to some other processes in the liver that are quite important for us, including detoxification, especially what has to do with the processing of the nitrogen, because nitrogen is of course a part of amino acids, and as a result of all the proteins from which we are all built. And thus, of course, we expect that there should be certain processes that involve the anabolism and catabolism of all these things, and we should get somehow rid of nitrogen because it's a little bit toxic for us. So we'll get to that in a while. So, but let's finish up with the usage of fats or fatty acids for getting some energy. So here is one more pathway of fatty acids degradation that has to do with so-called peroxisomes. So these are cellular organelles. So here is one on the right. So here you see that. And here you see some sort of crystal-like structures inside the peroxisome, so that's one way to recognize it. And peroxisomal oxidation, it differs from the beta oxidation in the initial dehydrogenation reaction. So here is that initial dehydrogenation reaction, and peroxisomes do not make ATP, and some energy is just lost in form of a heat. So I will show you some bigger scheme with comparison of here it is. I will show you that in a moment. Comparison of the oxidation in mitochondria and peroxisomes. But the thing is that you cannot effectively metabolize very long fatty acids with the mitochondria. So, but you don't want them to stick around and and just take some place in your cells. So you need some ways to metabolize them. So it's better to lose some energy in the form of heat, but anyway, perform certain reactions on that very long fatty acids. Besides, they can handle branched chain fatty acids. Very common example is so-called phytanic acid. So phytanic acid, I guess I will get to that at some point, but that's one of most important examples of the branched fatty acids that should be somehow handled inside of our cells. And it is handled with the process of so-called alpha oxidation. Some dicarboxylic fatty acids and some eicosanoids and unusual lipids can be also handled with the help of the peroxisomes. Okay, so what's the difference? Here you see some sort of comparison between mitochondrion and peroxisome or glyoxysome. And here you see the chemical reactions as they go and the difference between these chemical reactions. So here we start with certain fatty acids, or seeing this CoA, you guess it is already activated. So this is an acyl CoA. Once again, don't mix that up with acetyl CoA. So acyl CoA, it's some fatty acid connected to the cofactor A, so it has some long tail. Acetyl CoA means that you are just there are just two carbon atoms. Here can be a lot more carbon atoms. These systems, they differ in three aspects. So first of all, the peroxisomal system prefers a very long chain fatty acids. The second, in the first oxidative steps, electrons pass directly to oxygen, generating hydrogen peroxide. So here you see on the left the mitochondrial pathway. We are generating FADH2. This FADH2 goes to the respiratory chain or electron transport chain, and we get some ATP from the electron transport chain, and these electrons they are acquired with oxygen. So oxygen is the terminal acceptor of the electrons from the FADH2, but during these steps, I hope you remember that we will pump out some of the protons, we will get some proton gradient, and that proton gradient will be used with the ATP synthase to get some ATP for us. So pretty useful pathway. On the other hand, in the peroxisome, oxygen will acquire these electrons, but instead of water, hydrogen peroxide will be generated. Then the hydrogen peroxide will be transformed into water and some reactive oxygen species, and we will need to handle something with them. I guess this reaction it's probably performed with superoxide dismutase, but I'm not really sure, and I haven't written it anywhere, so probably that's the enzyme. Good. And the third, the NADH formed in the second oxidative step cannot be reoxidized in the peroxisome or glyoxysome, so reducing equivalents are exported to the cytosol and eventually are entering the mitochondria. The acetyl-CoA produced by peroxisomes and glyoxysomes is also exported, so they are not used immediately here as it can be in the mitochondria. And the acetate from the glyoxysomes, these are organelles found only in germinating seeds, so that's not in us. Yeah, and it serves as the biosynthetic precursor. The acetyl-CoA produced in mitochondria or acquired from the outside is further oxidized in the citric acid cycle. So here you see the reactions, how they go. So the first step we have considered, hydration, it's pretty similar. Here you see once again the difference, NADH that is formed in the mitochondrion, that's the path on the left, it goes to the electron transport chain, and once again we can acquire some ATP. In the peroxisome we get NADH, but there is no one to do something with that NADH, so it will be exported, eventually enter the mitochondria or whatever, and will be used for getting maybe some energy or whatever. The next pathway is pretty similar, but once again, when we get to the end, you remember that in beta oxidation we always get 2 carbon atoms at a time, and we are creating acetyl-CoAs, and these acetyl-CoAs can enter the Krebs cycle and give us some more NADH, FADH, and that will go to the electron transport chain and gain us some more ATP. In the peroxisome, you can do that, you don't have any Krebs cycle there, so it is just exported to the outside and then will be picked up with mitochondria or whatever process needs that acetyl-CoA. Here is a sort of coordination between fatty acid synthesis and breakdown. So here is the scheme. Well, you probably might guess that any biochemical processes, they should be somehow regulated, especially when it gets to energy, because the storage and activization or mobilization of energy from that storage, it should be somehow concerted with the amount of energy that we are able to get from the surrounding environment. So when we get some energy, it's time to store it for some future. If we are in a state we are not able to acquire some energy, so for some reason there is the food is scarce, or we are just waiting for some time to have a meal, so we can't eat something right now, then different signals they should start processes of mobilization of our energy storages. And here you see the scheme. So here you see the dietary carbohydrate. We can acquire some glucose from that, and depending on whether we have just eaten something or we didn't, we will get some signaling through insulin and glucagon. So insulin will tell us that we have just acquired some energy in form of, let's say, glucose, and it's really time to make some storages. So it tells to all the organs, guys, we have some high energy state. It's time to collect some glucose into your storages for some future usage. So use it when you need it, collect it, and so on. While glucagon tells us that there is the low blood glucose, and well, it's time to mobilize some of the storages. And here you see, so when the diet provides a ready source of carbohydrate as fuel, beta oxidation of fatty acid is unnecessary and therefore is downregulated. So two enzymes are key to the coordination of fatty acid metabolism. That's acetyl-CoA carboxylase, or ACC. So here you see it on this image. That's the first enzyme in the synthesis of the fatty acids. We will get to that synthesis at the end of today's lecture, I guess. And carnitine acyltransferase. So we have yesterday considered the carnitine shuttle, and that's the enzyme from that shuttle, and I told you that it's tightly regulated, and that's one of the points of regulation, especially for these sort of pathways. And here on the image, what do we see? So we see that ACC may be phosphorylated and thus inactivated, or it may be dephosphorylated and thus activated. So pretty common situation when you have certain enzyme, and these enzyme may be phosphorylated or dephosphorylated, and depending on that phosphorylation, dephosphorylation, it gets turned on or off. Many enzymes turn on when being phosphorylated. This one does not, so it's not some sort of strict rule. So depending on the enzyme, it may be turned on or off with the phosphorylation, and that depends on the exact enzyme you're dealing with. So what do we have here? So some, for example, we are in a state of high blood glucose. That triggers the release of the insulin. Now, this insulin-dependent some protein phosphatase dephosphorylates ACC and activates. ACC catalyzes the formation of the methanyl-CoA. So here we have acetyl-CoA, here we have methanyl-CoA. Where do we get acetyl-CoA? How do you think? One of the pathways we have studied with you contained acetyl-CoA as one of the final steps. So where do we get it? No, Krebs does not synthesize acetyl-CoA. It uses acetyl-CoA as source of basically cycling. But where does it come from? So what's on the other side of acetyl-CoA? Yes, glycolysis, absolutely correct. So when we start with the glucose, you remember we have 6 carbon atoms there. We process that glucose in the glycolysis, and at the end of the day we get to the acetyl-CoA. So there are different pathways when we finish the glycolysis, or when we finish that glycolysis, it can go to the anaerobic or to the aerobic pathway. So if we go the aerobic pathway, we get the acetyl-CoA. So that's one of the sources where we can get it from. And these acetyl-CoA, it can be used to synthesize the malonyl-CoA, and these malonyl-CoA can be later used for the synthesis of fatty acids. I will show you the exact pathway in when we get to that point in these slides, but you can guess that there are some steps. So we are something doing, and at the end of the day we get some fatty acids. You might even guess that this is some sort of cyclic process, so there is some sort of iteration when some malonyl-CoA is added to something, to some growing chain, and we are growing our fatty acid up to a certain point. So mostly something around 16 carbon atoms, but we'll get to that. The other thing is these malonyl-CoA synthesized from the acetyl-CoA, it inhibits the carnitine acyltransferase. So it inhibits this sort of transferase. That's the part of the carnitine shuttle that we have considered. So what does the carnitine shuttle? The carnitine shuttle teleports fatty acyl-CoA from the outside of the mitochondrion to the inside of the mitochondrion. There are some biochemical steps in between, so you remember it cannot just cross the membrane. There is no transporter that will take it as a whole, but there is a cunning way when some sort of acyl part is is disconnected from the CoA and connected to the carnitine, then this sort of construction can cross this sort of double membrane barrier, and then it is disconnected from the carnitine and reconnected to the CoA. But all in all, if you collect all the reaction, that looks like fatty acyl-CoA got from the outside to the inside of the mitochondria, and this part is inhibited. So if we have a lot of acetyl-CoA, we are inhibiting this part, and we are basically telling that we are in a high energy state and we don't need to mobilize our fatty acid resources to get energy. We have some better sources of energy, and there is no need for this part to work, because we already have some glucose, so we can immediately get ATP and so on. So we don't need these costly processes with many steps. And that's basically stopping the degradation of the fatty acids, and instead we are getting into the process of acquiring some new fatty acids through the way of fatty acid synthesis. And we will get to that in a couple of slides. Here are some nice pictures regarding the evolution of different enzymes. It turns out this is like sort of text describing that images and I wasn't able to fit that into one slide. But here's what you see. There are 4 enzymes of beta oxidation. For example, in gram-positive bacteria, they are separate soluble entities, and those are the short chain specific system of mitochondria. So here it is. So here you see these 4 enzymes. Here you see the description. So that's acyl-CoA dehydrogenase, anoyl-CoA hydratase, LB hydroxyacyl-CoA dehydrogenase, and the sialase. So here are these 4 enzymes that take the here is the fatty acid, you see, and they take it as an input, and produce some product. You see that they can be attached to a membrane. For example, here is the mitochondrial very long chain specific system, so how that looks like. In the peroxisomes, it looks like this. So here you see these four enzymes, and they are connected to the D3-hydroxyacyl-CoA epimerase and delta-3 delta-2 anylyl-CoA isomerase. So you remember we had some troubles with the double bonds in fatty acids, and we needed certain isomerases that were able to transfer that bonds into better positions, so that we can keep up with our beta oxidation. So these are two auxiliary enzymes, so these enzyme 5 and 6, and they all are parts of a single polypeptide chain. What's interesting, in mammals, all 7 activities are parts of a single huge polypeptide chain. So they are synthesized in a huge chain. How do you think? What are the pros and the cons of synthesizing some number of enzymes as a single polypeptide chain? So look at it, I don't know, like sort of engineering solution. So you are engineering some animal, and let's say as one of the problems, I told you that there is a system of several enzymes that are basically performing certain metabolic pathway one by one. Desired conformation, not sure. Oh well, no, I don't. I think there are some different reasons for that. Let's think about regulation. So you are studying how it's called, molecular biology, I guess. Yeah, so you should have been studying molecular biology, and I guess you should have studied some sort of regulation of protein synthesis or RNA synthesis. So can you think about that? Yes, they will be probably regulated with one promoter. So you are at once regulating all the proteins. The other thing is that you are ensuring that you are producing the exact number of copies of each proteins for like processing whatever you need in that pathway. So if you know that they will be processing something in chain, you will probably want the equal number of copies of each of these proteins. So if you are producing them as a one long chain, you will get that automatically. That's one thing. The other thing, you are regulating them with one promoter. That's a really good point. So if they were regulated with different promoters, it could have happened so that one promoter makes you to make a lot of copies of one of these enzymes, but the other promoter gets, I don't know, for some reason gets disabled or whatever happens to it, and you produce none to just a little bit of other protein. And since they all live in a one chain, in one sequence of biochemical events, that will probably stall the whole process. Because you know that everything goes forward only if you have, well, with the speed of the slowest enzyme. So we were considering that in our previous lectures, so we had certain pathways and we were searching for some bottlenecks in that pathways. So here it is. And what can be good if you create them as these as separate enzymes and regulate them with different promoters. Why didn't all organisms evolve to have them as a single chain? So some have, some haven't. So probably there is something good with synthesizing them as the separate proteins as well. Think about mutations. So if you have certain mutations and there is certain probability to damage something, which system will be more stable if you have just one single polypeptide chain regulated with one promoter, or if you have few different polypeptide chains maybe synthesized from pretty different parts of the DNA. So where would you get, let's say, a bit more stability? Yeah, absolutely correct. So if you have some more chains, if you have more parts, this system will be a bit more stable to mutations. So you can break something in one of the proteins, maybe it gets a little bit worse, but first of all the rest they stay intact. So since they are intact, you have still the same properties, and that one that is a little bit worse, well, maybe it will work somehow, maybe you will get some different copy of it somewhere. So you may have few different copies of that proteins in your DNA. So that may be pretty beneficial if you are in a state of some higher mutation rates or something like that. So that may be beneficial as well. So everything has its some sort of pros and cons, and this situation is not some sort of exempt from that. Good. One more biochemical pathway, that's the omega oxidation of fatty acids. So you might guess, if we are numbering our carbon atoms starting with the carbon next to the acidic group, there is a notion of the last carbon atom, so independent on what is the length of the chain, and that atom is called omega. So that's omega carbon atom. And in the endoplasmic reticulum we might get these process as well. That's an alternative fatty acid degradation pathway. So it targets this sort of terminal carbon atom. It prefers something like 10 to 20 carbon fatty acids, and under normal condition its importance is let's say minor, but it may become significant if beta oxidation is defective, for example in case of carnitine deficiency. Here you see the steps of this pathway. Like mixed function oxygenase adds the OH group to the terminal carbon atom. Then this is the group OH, so now this is an alcohol. So it's still an acid, but it has OH group, so it's an alcohol as well. And with an alcohol, we can involve the alcohol dehydrogenase that transforms that into an aldehyde. I guess I have mentioned alcohol dehydrogenase yesterday in the context of well, ethanol and methanol biochemistry, but here it works in the context of the omega oxidation. So here we have this sort of aldehyde group Now the aldehyde dehydrogenase. Dehydrogenase, you might guess, it removes some hydrogen, and here you see this group is transformed into the acid group, and then it may enter into the beta oxidation pathway. So here you see the result of this sort of processing. Good. What else? Phytanic acid or phytanic acid. I'm not sure how to read that. Maybe phytanic. That's a branched chain fatty acid. It is processed in peroxisomes. So here you see the phytanic acid. So here it is. Here you see these sort of branches. So like CH3 groups, and the thing is that we should somehow process that, but you might guess that beta oxidation on its own will not help us, because it is used to just a simple chain, and here we have some sort of branches in this chain. And the thing is, why do we need to metabolize that? The phytanic acid, you can find it in the typical Western diet. So whatever we eat typically will contain something like 50 to 100 milligrams of phytanic acid per day. So that may seem not much, but if we cannot metabolize that, if we cannot either remove it or use it for anything useful, that will be probably a trouble, because we don't want some garbage to be collected in our cells. So if they get something from the outside, they should have some ability either to remove that and get rid of that if nothing useful can be done, or use it for at least for something. So it has the methyl substituted beta carbon and therefore cannot undergo beta oxidation, and the combined action of the enzymes shown on the right. So here you see the whole chain of events of whatever is happening here. They remove the carboxyl atom, the carboxyl carbon of phytanic acid to produce prestanic acid, in which the beta carbon is unsubstituted, allowing the beta oxidation. And notice that beta oxidation of prestanic acid releases propionyl CoA, not acetyl CoA. So odd chain fatty acids, they release propionyl CoA. And there is even some sort of disease called Refsum disease that results from a genetic defect in phytanoyl CoA hydroxylase, and leads to the accumulation of very high blood levels of phytanic acid, causing still but by unknown mechanisms, maybe you will discover them at some point, who knows, some severe neurological deficits, including blindness and deafness. So that's a really big trouble. So if we cannot do something with that chemical, and in case of some enzymes are defective, we can't, that leads to some grave consequences. So you might guess that this sort of processing, it's really important. And here you see the pathway. So the synthase transforms phytanic acid into phytanoyl CoA. So here the acid group was changed to the this connection to the acetyl CoA. Then phytanoyl CoA hydroxylase adds this OH group here. Now the alpha hydroxy phytanoyl CoA lyase removes 4-mil CoA. So 4-mil CoA is removed and then processed, and that's what we left. That's a prestanol. So here it is. And now what we have, so this carbon atom is now closer to this change. And this is an aldehyde group. the aldehyde dehydrogenase transforms that into an acid. So here we have acid, and once again we perform certain manipulations with that acid, and we can process that. Good. So we have considered this pathway of getting energy. Now let's consider the pathway, what happens if we are in a state of low energy and we are scarce on some sort of glucose and some glucose resources. The problem is that we have certain critical tissues, especially brain, that need glucose, and there is no other way we can somehow supply them. So they don't have some fat storages, and they cannot store fat and then reuse it. We cannot transport acetyl-CoA to them. So there is a bit of a problem. What should we do? And there are different ways to tackle this problem. One of them I have mentioned for you yesterday, so we can use liver as the storage for glycogen that will be transformed into the glucose, and that glucose may be exported to the bloodstream and then reuptaken by the brain. But these resources are pretty scarce, I should say. So yes, we can leave some time bathing on that glycogen, but what happens when that glycogen is over? And it will be over much quicker than much earlier than we will get to the depletion of our fat stores, and that's pretty bad, I guess. So the nature supposes this way as well. So there should be some way to use fatty acids and our fat storages to keep our brain alive, because we still have enough energy to supply some rest of the organism and and keep it alive. And so it would be a waste to become brain dead in all these in this sort of situation. So there is a way, and this way is the synthesis of so-called ketone bodies. So we can synthesize ketone bodies that will be exported to the bloodstream, and then basically we will be able to supply some of these critical tissues. Here you see the recap, the glycolysis and glyconeogenesis. That's the synthesis of the glucose, but as far as we know, we cannot get from the acetyl-CoA to the glucose. So everything ends at the level of the pyruvate. So when we get to the pyruvate, we can still get back to the glucose and resynthesize and export it into the blood for supporting the brain. But if we are on the level of the acetyl-CoA, that's it. So no pathway exists in animals from acetyl-CoA into the pyruvate or whatever. And the problem is that fatty acids are giving you the exact acetyl-CoA. So there should be some other way so to do something. And here is that other way. So here you see the formation of so-called ketone bodies. And the ketone bodies, most cases we are interested in acetoacetate, D3-hydroxybutyrate, and acetone. They are formed from acetyl-CoA, primarily, of course, in the liver. So once again, liver is trying to support the brain so that we don't end up brain dead till we still have some energy. Well, and here you see these sort of steps. So one is performed with the keto thiolase. So here you see two acetyl-CoAs. It is transformed into the acetyl-CoA. So here is the formula. Then we get to the hydroxymethylglutaryl CoA synthase. Here it is once again some acetyl-CoA. It is attached to this sort of chemical. You get 3-hydroxy 3-methyl glutaryl-CoA, and then through the hydroxy methyl glutaryl-CoA cleavage enzyme, and you get the acetoacetate. So here it is, and now this acetoacetate, it can be either transformed into the acetone, or well, it can, let's say, spontaneously decarboxylate into the acetone, or under the action of D3-hydroxybutyrate dehydrogenase, it can be transformed into the D3-hydroxybutyrate. So here it is. These are basically the ketone bodies that are exported into the bloodstream, and that can be used with the brain to support well, its activity at least for some time. There's interesting part, if we have certain chemicals that are easy to evaporate or something like that in the bloodstream, they are pretty often found in the air that we breathe out. So that's a pretty common situation. That's the reason why when when police tests you for some level of of the ethanol in the blood, whether to check if you can drive a car or not, if they have certain thoughts regarding that, they are testing basically the air that you breathe out, and that air will contain some part of the ethanol that is correlated to the level in the blood. The same thing, of course, goes to the ketone bodies, especially the acetone. So if there is some level of the acetone in the blood, that can be detected in the air that you breathe out. So there may be some extreme situations when, especially when children, they use up their energy so vigorously that they get to the level of using up not only the whole storage of the glycogen, they are starting to use up these sort of storages of fatty acids, and the ketone bodies are started to synthesize, and that can be felt that something is not okay. So why the smell of the acetone? And the reason is basically that ketone bodies are generated and they get into the bloodstream. So that's part of some sort of metabolic process. Here you see some sort of scheme. So what's happening in here? So ketone bodies can be regarded as a water-soluble transportable form of acetyl units. So fatty acids are released by adipose tissue and converted into the acetyl units by the liver, which then exports them as acetoacetate and acetone. And as might be expected, acetoacetate also has a regulatory role. So high levels of acetoacetate in the blood signify an abundance of acetyl units and lead to decrease in the rate of lipolysis in adipose tissue. So there is some sort of regulation, so not to make them too much, not to make them too little, so something like that. So here you see how from the acetoacetate we can get to the acetyl-CoA. So acetoacetate, it can be dissolved in the aqueous, it can be transformed to aqueous solution, so it's exported into the blood and transported with the blood to everywhere. And if it is reuptaken by something, it can be transported to acetyl-CoA and used in the Krebs cycle, for example, by the brain. So here is one more scheme that shows the interaction of all the things in the organism. So here you see a fat cell. So I have shown you some images of the anatomy. Where do you find all these cells in the human organism? So it exports some glycerol, some fatty acids. Glycerol and fatty acids are uptaken with the liver cell. Anatomy of the liver we have already considered as well. Here are some biochemical mechanisms performed so you can transform glycerol into the glucose. That's very good. We can take fatty acids and transform them into the ketone bodies, and then these ketone bodies they are exported into the bloodstream, and they can be uptaken with different tissues. Especially what's interesting for us that the brain cell, the brain tissue. So here we have a brain cell. It takes up these ketone bodies, and with these sort of biochemical mechanism transforms that into the acetyl-CoA that is later used in the Krebs cycle to get some energy. So here you see these numbering and even some references. That image was, let's say, stolen from the Biochemistry of Stryer, so you probably know that book. Really heavy book, but I hope after our course you will like biochemistry so much that you will even read a few thousand of pages of that nice book. Good. So we have 30 more minutes. That's I guess that's really great. So here is some short info on the ketone bodies. So you see that major producers are the liver mitochondria. Then all that pathway I have described to you. The heart and renal cortex, they preferentially use acetoacetate over glucose. So even this can happen. So this tissue is pretty important for us as well, and you see that it needs these sort of ketone bodies. Brain normally uses glucose, but in case of starvation, or it may happen in case of diabetes, because in case of diabetes, you know that we don't have either enough insulin production or some defective receptors that detect that insulin. But whatever is the exact mechanism, the result is that some tissues think that we are in a state of starvation, despite the fact that some chemicals like glucose or whatever supplies energy are pretty abundant. So that's a large trouble, because first of all they are not metabolized correctly, and you get some much higher levels of glucose that you would like in the bloodstream, and that glucose may start attacking and glucogenating some proteins that should not be at normal conditions, and so on. So it's very very bad disease. So anyway, whatever happens, brain switches to getting energy from the ketone bodies, so to keep it somehow active during these periods of starvation. So what else? In the case of diabetes, there is seen so-called the diabetic ketoacidosis. Liver in case of insulin deficiency, or there is a case if some of the receptors are broken. So there are different possible mechanisms. So anyway, liver will not absorb glucose and will get insufficient of oxaloacetate, and adipose tissue will release some excess of fatty acids, thinking that we are in a state of starvation, and as a result we will get high ketone levels, so they will be transformed into ketone bodies, so the level of ketone bodies will be high. Here you see the exact numbers, so you might guess this is more than 30 times in blood, and here you see much more in urinary excretion, so that's really high levels. And of course that leads to the acidosis, because you have a lot of acid in the blood, and leads to many bad consequences. Okay, let's consider one more pathway that has to do not with the energy, but with some sort of managing the nitrogen. So we have a lot of processes that involve some nitrogen compounds. Especially we are interested in in amino acids because we are a form, let's say, of protein life. So probably we whatever we do, we are metabolizing some proteins, and these proteins are doing something for us, and so on. So we need some ways to manage waste that is created during the degradation of some proteins that we don't need anymore, or that are damaged for some reason and should be deconstructed. So that's the way. Uh, so during the normal synthesis and degradation of cellular proteins, some amino acids that are released from protein breakdown and are not needed for new protein synthesis, they may undergo oxidative degradation. Or when a diet is rich in protein and ingested amino acids, they exceed the body's needs for protein synthesis. The surplus is catabolized. Please note, we cannot store amino acids. You will have a homework problem that relies on this statement as well, so I want to make it explicit. So we cannot store amino acids. So whatever we have, we have. If we need more, we need to catch them and eat them. So that's the way it works. And during starvation or uncontrolled diabetes, when the carbohydrates are either unavailable or not properly utilized, some cellular proteins are used as the fuel. So some proteins can be used to get some bit of energy. And under all these metabolic conditions, amino acids lose their amino groups to form alpha keto acids, the carbon skeletons of amino acids. And the alpha keto acids, they may undergo oxidation to CO2 and water, or often more importantly provide 3 or 4 carbon units that can be converted by gluconeogenesis into glucose to fuel the brain, skeletal muscles, or whatever. And the pathways of amino acid catabolism are quite similar in most organisms. So everyone should do something with amino acids. But as you see here, it's not a big problem to metabolize the carbon skeleton. So it contains pretty regular atoms like carbon, like hydrogen, oxygen. You can transform that into pretty harmless carbon dioxide and water, so no problem. But there is nitrogen, and nitrogen is a little bit of a problem, so we need to deal somehow with that nitrogen. So here is an image that shows some ways of different amino acids and what can be done with all of that. So here is the liver cell. So here you see some amino acids from, for example, digested protein. So we have captured, I don't know, some animal and eaten that. Or that may be a result of degradation of some cellular protein. So here is the amino acid. Here is the pathway that transformed that into alpha keto acid. So here you see no nitrogen. That nitrogen is transported from to the alpha keto glutarate. You may remember this sort of chemical from the Krebs cycle. We had it in there. It is transported onto alpha keto glutarate to make glutamate, and then these glutamate may be created from glutamine, so whatever, and then we can generate ammonia, so NH4 plus, and it is later metabolized to something. So depending on the type of the animal, it may be metabolized to different things. Good. What I wanted to show you here. There are 4 amino acids. They play central roles in nitrogen metabolism. That glutamate, glutamine, alanine, and aspartate. So I'm not sure if I can show you them in these in this slide. Here you see a little bit more, so that's different forms of the nitrogen excretion. So here are, for example, urea, that's the chemical we use, and many other terrestrial vertebrates and sharks for some reason. In different animals, it may be excreted in form of ammonia as it is. For example, aquatic vertebrates, some bony fishes, larvae of amphibia, so that's the way. And here is the example of so-called uricotelic animals. That's uric acid used by birds and reptiles. So here are different forms of excretion. You might guess that if animal lives in the water, NH4 plus should be pretty soluble in that water because it has some charge, and water likes different polar or charged things. So it's probably easy to excrete as it is without getting into any troubles of synthesizing something out of it. While we have a little bit more troubles to synthesize something. Here is one more scheme. Here you see amino acids are formed. This is the pathway for the carbon skeletons. They get to alpha keto acids and then into the Krebs cycle to get on some energy. And here you see NH4 plus that should be somehow processed, including the urea cycle or ornithine cycle that we will consider, and that generates urea, the excretion product of the nitrogen. Good. Here is one more scheme, so of what is happening to all that chemicals. Here you have systemic circulation. So the liver is basically the only organ that is able to fully perform the ornithine cycle or the urea cycle. The cycle converts toxic ammonia, so NH3 is toxic for us, that is produced during the amino acid breakdown, the scheme I have shown you previously, into urea, which is safe and excreted by kidneys. Safe, but with some, I don't know, additional notions. This is not the best chemical to excrete, if being honest. The problem is the solubility in the water. So it has pretty low solubility, and the amount that we have in our blood is almost at that threshold. So if there is some problem, let's say maybe you have heard the disease of gout, if there are certain troubles, it may happen that these levels will be too much and it will start to crystallize inside of the body. So in case of gout, you maybe have seen how the fingers look, so it's pretty nasty disease, and that's basically the crystallization of the urea in that places, and it crystallizes there because most cases hands, fingers are the places in the organism with the lowest temperature, and you know that the lower temperature the lower that point of solubility, so the less of the chemical can be held in the solution, and it partially precipitates. So that's the case. So yes, it's like not toxic, but that's not the best choice. So probably if we will sometime get to the genetic engineering of the human, maybe we should reconsider this point and maybe add some more enzymes to produce something different. So it may be a good choice. The now what else? Two urea cycle enzymes are located in mitochondria of hepatocytes, and the other three in the cytosol. So the liver has the necessary oh my god. Compartmentalization. That's from the word compartment. Who knows why can't I say it? So probably we all are tired to the end of the semester. And the other tissue, they generate ammonia and send it to liver in form, let's say, of alanine. So we have seen this sort of cycle when we were considering the Quarry cycle. I showed you the pathway with the alanine. Or in form of glutamine, and liver detoxify that in the urea cycle. So here you see the example. So we have portal circulation, that we have eaten something, it gets to the liver. Here something is metabolized, urea is generated, part of it is gets through renal secretion, some gets into the intestine and through intestine secretion. The first step in the catabolism of most L amino acids, once they have reached the liver, is removal of the alpha amino group promoted by enzymes called aminotransferases or transaminases. And in many aminotransferase reactions, the alpha ketoglutarate is the amino group acceptor. So all aminotransferases have pyridoxal phosphate (PLP) as a cofactor. And although the reaction is shown here in the direction of transfer of the amino group to the alpha ketoglutarate is readily reversible, so its delta G is almost zero. So here you see the alpha ketoglutarate, here you see the L-glutamate, so here you see the new acquired nitrogen atom, and here you see the another amino acid, so these R signifies that it can be pretty arbitrary amino acid, and this is the nitrogen atom that was abstracted and transported to the alpha ketoglutarate. So here you get some alpha keto acid, so it's not more amino acid, it's just keto acid. So here it is. The logic may be pretty simple in this case. Let's say if you have many amino acids and you should do something with them, so there is some critical catabolism that should go. It's pretty inconvenient to create 20 different pathways for each of the amino acids. So you might guess that it is pretty reasonable. If the trouble is the nitrogen atom, so why don't we take that nitrogen atom to just one chemical and then we create a pathway for doing something with that chemical. So that's pretty reasonable. So we don't need 20 different pathways, we have just one, they are converging into one pathway. So I guess you will see that in biochemistry some more examples of that. So here you see this sort of mechanism. So all aminotransferases, they have the same prosthetic group, the PLP. The coenzyme form is the vitamin B6, so vitamin B6 is needed for that. The pyridoxal phosphate function as an intermediate carrier of amino groups at the active sites of aminotransferases. So you know that reaction can go here or there, so delta G is almost zero, so it just keeps for some time that nitrogen atom to get rid of it when the next chemical comes. So here you see that that's called the ping-pong mechanism. So here it is in the Cleveland notation. So some chemical gets attached, some product is generated, then a different chemical is attached, a different product is generated. Here is a more traditional scheme that shows some intermediate versions of all of that. So here you see the enzyme and substrate 1. They create that sort of enzyme substrate complex. Reaction goes, you have product 1, but now enzyme is modified, so that's enzyme prime, so primed enzyme, so it has changed somehow. And these enzyme primed may interact with substrate 2, that leads to recovery of the initial state of the enzyme and generation of the product 2. And here are some more details on the biochemical mechanism. So here you see the pyridoxamine phosphate and pyridoxal phosphate, PLP. So what happens here, you see here is the nitrogen atom is attached, here is no nitrogen atom. So on the enzyme you have the lysine with the nitrogen atom. It can attack, so nucleophilic attack for this sort of carbon atom in the PLP, so that's the prosthetic group, that's the PLP, and here you have the Schiff base is formed after this reaction. So here are some more reactions. I guess since we are so scarce on time, I guess I will not get through all of them, but you can later look what happens here. So that's reactions of transamination, racemization, and decarboxylation. So all they involve this sort of prosthetic group, the pyridoxal phosphate. So here it is, and here you see what happens in different cases. Here a amino acid is attached, creates some sort of Schiff base, then another base abstracts a proton, so reaction goes, and here you have pyridoxamine phosphate, and here is the alpha keto acid that is released. This process, the process A, should go one direction and then all the way back. So in one direction it takes away this nitrogen atom from the acid, amino acid, transform that into alpha keto acid. In the other direction, some different alpha keto acid may come here and get back the nitrogen. And here you see the some other let's say reactions that are performed with this sort of PLP. Here you see the reminder regarding the glucose alanine cycle. So here you see the in the muscle you have glucose undergoes glycolysis to the pyruvate, and some muscle proteins may be degraded in some cases, and at the end of the day it is transformed into the alanine that is transported through the blood to the liver, and the liver performs some more chemical reaction, like transforming that back into the pyruvate, into the glucose, and exporting the glucose that can be used by different tissues, while this alanine, as I hope you remember, that's amino acid, and we have part of it transformed into the glucose, but how do we get rid of the nitrogen? And so here is the way. So alanine aminotransferase transport that nitrogen to the alpha ketoglutarate, and that goes down. Interesting point to notice, this alanine aminotransferase, or abbreviated ALT, is one of significant clinical let's say measurements for the state of the liver. The thing is, if you ever measure in the blood some proteins that are supposed to be inside of cells, not outside of them, that tells you that something is wrong. So some cells, they are, for some reason, they are damaged. So in case of liver, so-called ALT and AST, that's alanine aminotransferase and aspartate aminotransferase, these including also some sort of some phosphatase, I guess I have forgotten the name, and some other enzymes. If they are detected in the bloodstream, that means that the liver is damaged and the things are going not so good. Here is the reaction catalyzed with the glutamate dehydrogenase. So here you see the reaction. Here is the glutamate and how it gets to the alpha ketoglutarate and gets rid of the nitrogen atom. These enzymes of mammalian liver have the unusual capacity to use either NAD plus or NADP plus as the cofactor. The glutamate dehydrogenases of plants and microorganisms are generally specific to one or the other. So here you see that in the image, so or either NAD or NADP. Good. What else should I show you? Yes, ammonia transport in form of glutamine. So excess of ammonia in tissues is added to glutamate to form glutamine, a process that is catalyzed by glutamine synthase. And after transport in the bloodstream, the glutamine enters the liver. So once again, the liver, NH4 plus is liberated in mitochondria by the enzyme glutaminase. So here you see the process, so a glutamine synthase. So here is L-glutamate. Here you see some unit of energy in form of ATP is consumed. You get gamma glutamyl phosphate. Then you attach that nitrogen atom, so this NH4 is attached here in place of the phosphate. Here you have alpha glutamine, so it has two nitrogen atoms. So one it has just because it's amino acid, the other one is attached here, and then it can be transported to somewhere, well, basically to the liver, and in the liver and especially in the mitochondria it may undergo some process. It will be processed by the glutaminase. So here you have L-glutamate at the output, and NH4 is liberated and can undergo certain processes that will transform that into the urea. So that's so that's it. You see, the problem is that ammonia is pretty toxic, especially to the brain, and in blood, the 98% of ammonia is in form of NH4 plus, which cannot cross membranes, and the small portion in form of NH3 can cross the membranes, including the blood-brain barrier. It will accumulate in cells and cause the troubles. So NH4 plus is a little bit better than NH3. You might guess why. NH4 plus has a charge, so it cannot cross the lipid bilayer, because lipids are hydrophobic. They don't like anything charged or really polar or something like that. NH3 has no charge, it's a small molecule, so it can cross. So that's the problem. It's better to keep it in form of NH4, or even better to process that in the liver into something better. So here you see the ways it disrupts the brain, so like problems with potassium homeostasis and so on. I don't think I will get into all these details. You may look up them a bit later. Here is basically the urea cycle, or the ornithine cycle, that I have promised to you. So for big steps. Here it is in the image with the compartments shown in here. So that's mitochondria, that's a cytosol. So parts of the reactions are performed in the mitochondria, some reactions are performed in the cytosol. So the ornithine transcarbamylase, that the step number 1. So here you see the ornithine, here the carbamoyl phosphate that comes here, and here a citrulline is created. So here you see all the different pathways that bring nitrogen atoms here. The NH4 plus is synthesized, and through the carbamoyl phosphate synthase, a carbamoyl phosphate is synthesized. So here you see the reactions, how that goes, and how that is transformed into the citrulline. So here you have that new hydrogen atom acquired. Good. The citrulline is then exported into the cytosol, and here you have arginine succinate synthase that performs reaction, creates citrulyl AMP intermediate. So here you see the AMP part and adenine monophosphate part, and here you see that we have that citrulline. Then it undergoes the step B, that's the same arginine succinate synthase that creates arginine succinate and liberates AMP. So here it is. And now the third step that basically by arginine succinate in the cytosol once again creates arginine. So here you see this arginine. It has a lot of different nitrogen atoms, and at the end of the day we have arginase that generates ornithine. So ornithine is generated from the arginine, and a urea is generated. You see it carries two nitrogen atoms, and these nitrogen atoms they will be excreted through either renal excretion or through the intestine. So that depends. Good. I guess I have used up all my time. Then let me just check. Let me just check. Good. I guess I just say a few words, and I will skip the synthesis of the lipids. I will leave them for you as some sort of part of the homework. So here are different links between urea cycle and citric acid cycle. So here we have citric acid cycle, and here we have the ornithine cycle, and so on. So these are just some sort of links. Here are these ALT, AST that I have told you, and other enzymes that have certain medical relevance. I guess you can check that to get some idea. Once again, some summary of amino acid catabolism. So where do they go, and what can they be transformed into? Just a small notion that some of them are called glucogenic. So here they are. Here's the list, and they can be transformed into the pyruvate, some other into the oxaloacetate, fumarate, succinyl-CoA, or alpha-ketoglutarate. So they can enter either citric acid cycle, or they can do something. The oxaloacetate, it can be used for the gluconeogenesis later. Yes, you might want to think that's a tricky question for you, for like sort of homework. So why can we make from oxaloacetate, we can return back to the glucose, but what's the problem with the acetyl-CoA? So why can't we use that to generate some new glucose? So you may want to think about it. Good. These are just some small reactions. So liver performs the heme degradation. So our red blood cells, they don't live for a very long time. So we need somehow to degrade whatever's inside, and heme degradation is one of the pathways that are performed in the liver. And maybe you can guess this cute guy. He's referencing to which one of the chemicals in here, in this scheme. Does anyone know? No? That's reference to the bilirubin. If you have ever read Silence of the Lambs, you may know that. So I'm not getting into all of these details, pretty detailed, but Uh, Good. And lipid biosynthesis, I will probably leave for you as some sort of homework, that just to look at these nice reactions, how they go, and how do we synthesize all these nice lipids and bile salts as the part of that lipid biosynthesis. Good. With that being said, if you have any comments or questions, please fire away. I guess no comments, no questions. Good. Then have a nice evening and see you next week.